Yeah, welcome to it.
And hopefully you all can hear me okay. I might mute my video at some point if the internet connection gets shaky. So we have 3 students who will be presenting their papers this morning until 10:50. And what I'm going to do is introduce each student before their paper presentation starts. And then we will have 15 minutes for Q&A at the end, around 15 minutes of time for Q&A at the end after all 3 students have gone. So coming up first, I would like to introduce Jenny Lewis Francisco, who's a junior at the University of North Carolina at Greensboro, who is majoring in sociology and environment and sustainability. She is also an honors student at the Lloyd International Honors College. Jenny is also the editor for Idre Goch, and I did not pronounce that right, an interdisciplinary honors journal, which was launched by the Lloyd International Honors College that features submissions from students across diverse academic disciplines. So welcome, Jenny, and you have, I think, up to 10 minutes, so I will turn my camera back on when we are getting close to that time.
Awesome, perfect, thank you. Let's see. I would like to share my screen. If
that would be helpful.
Jenny, I'm making you a co-host, so you'll have access to do so.
Perfect, thank you.
Thank you, Eric.
I'll do so for the other presenters as well.
Awesome. Let me quickly set this up.
So they're doing all use Zoom. They're not really speaking to an audience.
Right, it's a little bit different.
Okay, awesome. Well, good morning, everyone. I just want to thank the session chair for taking the time to moderate the session, but also the student presenters for sharing their research and their hard work. And for my specific research, I wanted to talk about the importance of inclusion and diverse voices in environmental literature. And I don't want this to be another lecture or PowerPoint about the importance of inclusion and diversity, but I wanted this to be a I do have a couple of questions that welcomes conversations and deliberations about this topic, and you're welcome to answer. You're welcome to answer in the chat or unmute for a few seconds. We do have 10 minutes, but a lot can be done in 10 minutes. But I do ask that we have respectful dialogue when answering these questions. But it's also a brave space for you to share. And in 30 seconds or your answers in the group chat would be great. But before we get started, I wanted to talk about the thesis of my paper. And the main question I was trying to answer was what makes diverse voices in environmental literature so unique? And if I could find this answer, I will be able to understand why we need diverse authors, and especially in environmental literature. And among all the most read environmental books from Rachel Carson, John Muir, that have expanded our appreciation and care for the natural world, there are hidden stories, hidden knowledge that are yet to be heard. From Indigenous people's scientific understanding to the different viewpoints from marginalized communities, these authors have the opportunity to kind of expand our understanding of the environment and call for action with sustainability. And there is a need for more diverse voices, not only for that aspect, but because authors of marginalized communities and other disproportionate represented communities, they have a way of intersecting elements of the environment and recognizing the different interconnectedness of social equity, the environment, economics, and aesthetics. So the first question, again, you're more than welcome to write in the group chat or unmute for a few seconds. And that is, what are environmental literature pieces or books have you read that are impactful to you? And how did that shape your view about the environment or sustainability? Again, you can feel free to unmute for a few seconds or write in the group chat, which I'm not sure if I could see. Let's see. I'm not able to see the responses, but afterwards I'll definitely take a look.
If it would be helpful, I can read them out if we have any.
Yeah, that would be great. Thank you. So with that being said, if you reflected on these questions, in this paper, I compared 4 different authors of environmental literature, one from John Muir, one from Robin Wallkimmer, a scientist and a member of the Potawatomi Nation. And we also have Bill Gates and also Mathai Wangari, who's from Kenya, and she is a social and environmental activist that has done so much great work. And so for the beginning of this research that I did, I wanted to see the difference of how their approach to their relationship with environment. And with John Muir's, the famous preservationist, famously wrote Hetch Hetchy, in which he writes about the dangers of the valley that is in danger of becoming a reservoir for San Francisco citizens to use for electricity. And he uses descriptive writing and rhetorical devices kind of to emphasize the aesthetics of the valley. That way, readers can feel like they're in that valley and feel the need to preserve it. He uses that approach, but the shortcomings of it, he doesn't necessarily call for a holistic, sustainable solution for maintaining or caring the environment, or even providing a social equity aspect for those who reside in the valley, or alternative sustainable solutions for citizens in San Francisco. Instead, he focuses on kind of the use of aesthetics to focus on the dualism of that distinguishes people and the environment, instead of encouraging sustainable approaches or preventing environmental damages or protecting communities. But contrary to that belief, we have Robin Wallkimmer. You can see from the PowerPoint at the bottom, one of her quotes in her book, Braiding Sweetgrass. Contrary to Muir, she actually uses, she believes that there is a reciprocal relationship between people and nature. How she dives deep into indigenous people's wisdom, their knowledge, their scientific knowledge, the history of colonialization, the restoration efforts led by various indigenous tribes, and even botany to explore that connection between nature and kind of human efforts. And I added this quote from her book, which I thought was just really transforming in how I kind of perceived the environment. And I really want to read this quote. And she says, "We are linked in a co-evolutionary circle. The sweeter the peach, the more frequently we disperse its seeds, nurture its young, and protect them from harm. Food plants and people act as selective forces on each other's evolution, the thriving of one in the best interest of the other. This to me sounds a bit like love." And even just from that piece, Kimmer writes that nature, they are gifts, and in Potawatomi culture, to show that gratitude, they show gratitude through reciprocation. And she wants readers to realize that we have to treat nature as a loved one. And when we do, we are able to think of sustainable solutions for future generations to come. And the next question kind of moving on to the beliefs and values of the environment, and what are your values and beliefs about the environment, sustainability, and what shaped your values and beliefs? Again, you can post your answers, but I really want you to reflect on it. And for these next 2 pieces, I looked into Bill Gates on his recently published book, and Wangari Maathai. For these 2 pieces, they focus more on sustainable action. But I feel like for Bill Gates, he emphasizes, again, investments in technology, public policy as primary sources of sustainable solutions. And, he crafted his message for this book to people who are in position to invest with innovative technologies to create policies and invest in clean energy. And he kind of limits his audience to those who are able to. Granted, he is his his expertise lies in economics and technology. But to spur readers to take action is to address the interconnectedness of social equity of the environment, economy, and aesthetics. And I think Wangari Maathai really does a phenomenal job with engaging readers to these elements. In her book, The Green Belt Movement, she talks about kind of the how to use philosophy, goals, and specific steps to create grassroots environmental movements, organizations that are founded on social justice. And she uses her personal experience, she uses images, she uses her lived experiences from the collectivity of the women she worked with. And she really uses that to combine science, history, and culture and overlap it with social equity and the environment and economy and aesthetics to really create change as a result of planting millions of trees in Africa. And I think, while Gates focused on solutions that are accessible to the privileged, Maathai focused on collective action. And just to wrap this up, again, all these authors that I've noted, they all played an essential role in the environmental movement. But there are different lived experiences, there are different messages that can be sent across. And inclusion in environmental literature can expand viewpoints, can expand approaches to sustainability. And these pieces have used aesthetics kind of to identify these issues. But yeah, there are many environmental challenges that require creative and collaborative actions. But how can we make this meaningful change if books are limited to a selected few? And as Kimmer lastly pointed out, we can't meaningfully proceed with healing, with restoration without restoration. In other words, our relationship with land cannot heal until we hear its stories, but who will tell them? And I think there are much more stories beyond these two women that I've talked about and the experiences to learn from. And all we have to do is take the time to open the book, open their books, open their stories, and to listen and to read. But yeah, thank you.
Thank you, and a big round of applause for Jenny. Alrighty, and a reminder, we'll do Q&A at the end for all three students. Our next student who will be presenting is going to be Natalie Sevistone.
Yes, Natalie Sevistone.
Sevistone, thank you, who is a senior at UNCG graduating in spring 2022. She is pursuing a Bachelor of Arts degree in Biology and Environmental and Sustainability Studies. So Natalie is also a member of the UNCG Lloyd International Honors College and has been an intern for Dr. Kerner's ecology lab at UNCG for a year and a half. All right, take it away.
Sure. This one. Yes, so hi everybody, I hope you're all doing well today. My name is Natalie Sevistone and I'm double majoring in Environmental Sustainability Studies at UNCG. For the past 2 years now, I've been working in Dr. Kerner's Biodiversity Ecology Lab on campus, and for a year of that, I've been conducting my own research within the lab. And today I will be presenting that research entitled Impact of Nutrient Deposition on Arthropod Communities of the Longleaf Pine Savannah Ecosystem. So in this presentation, I will begin by giving a brief background on the Longleaf Pine Savannah ecosystem and its history, and then I'll share my initial goals and hypotheses of my research and go into explaining my methods that I used in order to address those goals as well as my results. So there are many reasons why the Longleaf Pine Savannah, the Longleaf Pine Savannah is home to many different plants and animals. It's a very diverse ecosystem, and as you can see on the map to the right, it historically existed through a large part of the southeastern United States. It covered about 37 million hectares of the southeastern United States. Currently, it only covers about 2.2% of that original area, and due to this loss, a lot of the original biodiversity has also been lost, and many of those plants and animals are now endangered. So despite this destruction, the longleaf pine savanna still contains valuable and diverse plant and animal life, which is largely found in the understory. And you can see an example of that to the right. The understory is the area that exists below the canopy or beneath the trees. And as you can see from the picture, it mainly consists of grasses and flowering plants in the longleaf pine. So there are a few reasons as to why the longleaf pine savanna is currently threatened. It all began with European settlement. The ecosystem provided these people with many resources such as timber, tar, and turpentine. They took advantage of all of those resources, and it led to an overexploitation of the ecosystem. Deforestation, in order to get those products and resources from the 1700s to the early 1900s, led to the decimation of the ecosystem. And additionally, the longleaf pine savanna is dependent upon natural forest fires. So the understory grasses make sure that these natural forest fires are spread evenly across the floor of the longleaf pine, and it does so in order for the pine to maintain its health and biodiversity. So for example, research by Brockway et al. In 2005 showed that a certain level of fire disturbance is ideal in order for dead tissue and debris to be burned away throughout the understory of the longleaf pine, and new plants are able to grow and replace the old plants that existed, and ends up being an increase in resources after these natural forest fires go throughout the ecosystem. So therefore, decades of fire suppression has been detrimental to the longleaf pine savanna ecosystem and the health of its biodiversity. Climate change has also caused changes in the composition of the ecosystem. It has created more frequent heat waves, and nutrient levels and availability are also expected to change in addition to this, and that's actually what my research today focuses on. This can influence global nutrient cycles, which impacts nutrient dynamics in terrestrial ecosystems. The above ground productivity, or known as the biomass of plants, is limited by nutrient availability. And if it's negatively impacted, then this will also affect how natural fires are distributed across the floor of the longleaf pine savannas. All of these factors can influence the native and highly diverse plant and insect communities within the longleaf pine ecosystem. And while the plant communities have been extensively studied, the insect communities have not. And it's important to focus on the insects as they provide numerous services such as pollination. And as you can see in the previous picture, there are many flowering plants in the longleaf pine. This all means that we need a better understanding of the effects that changes in resource availability and nutrient availability has on insect abundance and their communities. And just to reinforce the point that insect and plant communities do go hand in hand, so considering the impacts that they will have on each other is very important in future restoration efforts for this ecosystem. So I began my research by asking these initial questions. The first being, how does nutrient addition affect insect abundance? And the second being, how does this affect insect community composition? I predicted that because nutrients can affect the physiology and limit the growth of insects, insect abundance will be greater in all of the nutrient addition plots when compared to the control plots. And insect community composition will be different, significantly different between the treatments. So this experiment was completed within an already established experiment. And the established experiment is in the Sandhill Game Lands of Hoffman, North Carolina. It simulates nutrient deposition in order to see how future nutrient availability will affect plant communities specifically. And it's part of a global network of sites that are conducting the same experiment. But I personally wanted to focus more on the arthropods that also existed within these plots rather than the plant communities that were already being researched. I took trips with other students from Dr. Kerner's lab to Hoffman, North Carolina, as you can see in this picture. And that's where part of the longleaf pine savanna does exist in North Carolina, and the nutrients were spread evenly among many different plots in this area. So this next picture will show all of the plots that exist within this established experiment. So in this picture, there are 30 5-meter by 5-meter plots. There are 3 control plots and 3 droughted plots. Plots that receive nitrogen or phosphorus or potassium or different combinations of those 3 nutrients. And then there are 3 replicates of each. But my specific research only focused on these specific plots. It's a subset of the ones in the other picture. These plots have the specific treatments that I'm focusing on, P being phosphorus, K plus being potassium, N being nitrogen. And there are still the individual nitrogen, phosphorus, potassium treated plots, and then various combinations of those 3 things. These study plots are also 5 meters by 5 meters, and there's a 1 meter buffer in between each plot. Just to continue the methods, when we went to the field site, arthropod samples were collected by using a modified leaf blower. So instead of blowing out, it sucked them all in. Insect samples were then stored in coolers at the field site until they were taken to UNCG, and then they were stored in the freezer until they were ready for identification. Using a microscope, I identified the individual arthropods and I classified them to their family grouping. As you can see in this picture, it shows the different groupings. I wrote down their order and their family. There are some pictures of a few insects and a spider that I actually identified in the lab. The 3 pictures on the bottom were just taken by my phone, putting it through the microscope lens. The one on the top left of the jumping spider was actually taken on an Amscope digital camera that I attached to the microscope. And it helped take some better quality pictures. And it was a part of my protocol, which allowed me and other students to easily identify these same insects in the future, because we could go back, we could refer to these pictures, and it would help move the process along a little bit better. And as noted, thank goodness these insects are not to scale. So the following analysis methods were used in order to create graphs and provide statistics for the data collected, and all of this was done in the statistical program R. In order to compare total arthropod abundance across the different treatment plants, I used an ANOVA, which stands for Analysis of Variance, and it's used to look for differences between the means of 3 or more independent groups. Here, I wanted to determine if across different levels of treatments, is there a difference in abundance, and is this difference significant? The second data analysis method was Permeanova, and that was to compare arthropod communities across the treatment plots. It stands for Permutational Analysis of Variance. And in this specific test, I used the Bray-Curtis dissimilarity. I calculated that, and it's used to determine how different communities are from each other across the treatments. And finally, I used an Nmds Plot, and it is a multidimensional space that depicts how different these communities are from one another, and it allows us to visualize the communities, and I'll show an example of this later on in the presentation. So this graph shows order richness, or how many specific orders were present. On the y-axis, you can see the order richness. On the x-axis, you can see the different treatments. You can see from this graph that phosphorus, or P, and potassium, K+, plots in dark blue appear to have more different kinds of arthropod orders, which with most nutrient addition plots having more different orders when compared to the control plots. However, the potassium plots in orange were the least diverse, even less diverse than the control plots. However, when that potassium is combined with either nitrogen or phosphorus, then those plots are more diverse than the other treatments. Despite seeing these trends, these results are not significant, which for now indicates that arthropod order richness is not influenced by nutrient additions. Similar results can be found when looking at this plot. It shows family richness. So family is a more specific identification for the insect, so it provides a more detailed description of arthropod community diversity. This graph shows family richness, or how many families were present, shown on the y-axis, and again, the treatments are on the x-axis. The phosphorus treated plots are more rich than the control plots. However, N and potassium plots were less rich compared to the control plots. And what's interesting here is that when nitrogen and potassium are combined, once again, either with each other or phosphorus, then they are more diverse than the control plots. It's also not significant, as is this one. This graph shows the total arthropod abundance. So it's just all the arthropods together. It's how many of each order. You're not looking at specifically order of family, just total arthropod abundance. The phosphorus plots have the greatest arthropod abundance, despite not having the highest diversity levels when you look specifically at order of family. The potassium plots have a lower arthropod abundance compared to the control, which is a recurring theme, but when combined with another treatment, the abundance is greater. This graph shows abundance once again, but it splits up the columns by the orders. We can see that phosphorus may have a greater total arthropod abundance, specifically because it has more insects identified as Diptera or Columbella than the other plots. It's also interesting to note that nitrogen and potassium treated plots have no Diptera, which indicates that the nutrients in these treatment plots may affect overall arthropod community composition of the plots. And finally, this Nmds plot, as mentioned previously, it plots the arthropod communities belonging to the different treatments in a multidimensional space in order to visualize how different these communities are from one another. So in this plot, if two colors are closer to, or 2 points at all are closer together, then that means that the communities are more similar. And if they're farther away, then they're less similar. This plot actually did show a significant difference between arthropod communities. The p-value was 0.02, which is a significant value. And it's interesting to note that even some communities of the same treatment can still be far away from each other. So if you can tell, the colors are a little bit similar, but the two on the right are both nitrogen, and they're actually very far away from each other, even though they're both treated with nitrogen. So just to finish it off with a little discussion portion, it does not appear that nutrient deposition will affect communities, insect communities at this time, but further research must be done. I was able to conclude that the arthropod communities are significantly different from each other between each treatment. It's possible that what's driving this difference is that nutrient deposition are changing the plant communities as a whole, which in turn influences the arthropod communities. Nutrients could be affecting the grasses, and the grasses may be taking over instead of, say, the flowering plants, so then different arthropods would be attracted, such as pollinators would be less attracted since there are fewer flowering plants. The way in which insect community changes over time with various nutrient treatments will inform future restoration practices of the longleaf pine savanna ecosystem, and it's important to consider the impacts that insect herbivory will have on plant communities of longleaf pine savannas as both communities do coexist together. Global temperatures will also continue to change, as we know, and nutrient levels and availability are expected to change along with it. This can and will influence the global nutrient cycles, which impacts nutrient dynamics within terrestrial ecosystems. And finally, investigating how plant and arthropod communities respond to changes in nutrient levels will lead to a better understanding of these communities as they relate to the longleaf pine savanna ecosystem. So here are my citations, and I want to thank you all for coming, and I guess we'll answer questions at the end. So that's all I have for you guys.
Thank you. Alrighty, I left the data visualizations personally. I work with those a lot in my day job. Alrighty, so our third student is Zachary, who is currently a biology major at UNCG in their senior year of study. Zachary is an international honors student at the Lloyd International Honors College and an undergraduate researcher in the Kerner Biodiversity Lab as well. And whenever you are ready, Zachary. And you are muted.
Give me 1 second.
Ooh, cool.
I'm sorry, I was muted. Can you see that okay?
Yes, but I am seeing not the presentation mode.
Okay, how's that?
And now I see the presenter view.
Okay, hold on.
All right, cool, now I see the presentation.
Awesome, perfect. All right, so my name is Zachary Bunch. I'm currently a senior here at UNCG pursuing a biology major and a chemistry minor. I've been an undergraduate research for about 1.5 years, and I currently work in the Kerner lab, which has a focus on the factors impacting biodiversity. I'm excited to share my project, the impacts of invasion on the insect communities of Montana rangelands with you all today. So there we go. So first of all, what is invasion and why does it matter? An invasive species is a species that is introduced to an area and causes net negative harm to an ecosystem or an economy. Some species, such as the European honeybee, are non-native but aren't necessarily considered invasive by the ecological community due to their agricultural and ecological benefits, as well as lack of significant disruption to native species. Our grass species for this experiment, Bromus arvensis, is considered to be an invasive species because it decreases forage quality and negatively impacts plant diversity or rangelands. The threat from invasive species is present virtually everywhere in the world, plaguing nearly every ecosystem on the planet, from streams to wetlands to forests to grasslands. Some examples you may be familiar with are the Burmese pythons in Florida, as well as the invasive murder hornets first spotted in Washington. These examples are species that are highly aggressive predators. As you can see from the picture on the right, are quite large and quite frankly scary looking. This size and aggression means their impacts are very immediate and measurable. So what are grasslands and why do they matter? A grassland is an open and expansive area of land with continuous or near continuous grass coverage throughout. While vegetation coverage is primarily grass, these ecosystems are still incredibly diverse. As you can see in this figure, grasslands are very common with coverage on every continent other than Antarctica. Rangelands are grasslands that are used for grazing livestock. These rangelands are very important as they are central to the global meat supply. Additionally, some farmers will graze sheep, dairy cows, or goats to enhance global wool and milk supply. Therefore, protecting these rangeland ecosystems has benefits from both a conservation angle as well as a financial standpoint. So why are insects important and why should we study them? Insects serve a number of very important roles, especially those related to ecosystem services. Some examples include pollination, nutrient cycling, and decomposition. However, insects herbivores also compete directly with livestock for forage. During the growing season of Bromus arvensis, our invasive species in study, insects and cattle avoid it and opt for native forage instead. This means that there may be increased pressure on our native forage, which could negatively impact cattle productivity, as these native forages are the plants they prefer. In a broader sense, insects are also vital to the overall functioning of an ecosystem, and therefore should be examined to understand the full scope of the impact of invasive species. On this slide, I have some background information on our collection site for this study. For this study, we are focusing on rangelands in Montana, where the previously mentioned invasive grass species, Bromus arvensis, is widespread. On the top left, we have an example of a research plot that was highly invaded by the grass species Bromus arvensis, which is shown more closely in the top right photo. On the bottom left is an image of cows at our study site at Fort Keogh, Livestock and Research Laboratory in Montana. I included this picture just as a reminder that Fort Keogh is an active rangeland in which cows constantly graze. On the bottom right is just a picture of the entrance to Fort Keogh, but it gives you a feel for what this location is like. Essentially, this area is not only actively used for ecological research, but also serves as a working rangeland that's important for biodiversity, livestock production, and many other ecosystem services. But once again, it is all threatened by invasion. So this brings us to our research question. How are insect communities in Montana rangelands affected by invasion? In this case, the presence of Bromus arvensis. So sample collection took place in the summer of 2021 using a modified leaf blower from plots indicating a 0, 50, or 100% level of invasion. The samples collected were then transported from Montana back to our research lab. There, we sorted the samples by separating the plant debris from the insects. The saved insects were then identified to order, with a total order abundance count being taken for each plot. For clarity, when I say order, I'm referring to what is known as a taxonomic order. I placed a graphic on the lower right-hand side that may be familiar to some of you. A taxonomic order is broader than a family, but more specific than a class. The order data we collected was then analyzed to determine the differences of abundances across different levels of invasion. On this side, I have some examples of what we found. These photos definitely took some time to get, as we had to use a microscope camera and lots of focusing and setting adjustments to get the clarity you see here. On the top right, or on the top, we have what you may immediately recognize as a grasshopper, which is part of the Orthoptera order. On the bottom left, we have an ant, which is part of the Hymenoptera order. On the bottom right, we have something interesting. We have what we believe to be insect eggs. Unfortunately, after contacting several people, we concluded that without the ability to hatch these eggs, we would not be able to identify them, but they were included as unknown insects in our abundance measurements. Moving on to our results, in figure 1, we have invasion percent or abundance on the x-axis with the abundance of each insect on the y-axis. As shown in figure 1, average total insect abundance is highest at the 0% level and lowest in the 100% level. Each color represents a different order, which is a taxonomic classification above families. You can see the most visibly abundant order is Orthoptera, which includes numerous families of grasshoppers, locusts, and crickets. Arachne is also a major order and includes families of spiders. Hemiptera is also very important in a major order as a driver of plant herbivory along with Orthoptera. Here we have average insect abundance at each invasion level on a box plot. On the y-axis, we have insect abundance, and on the x-axis, we have invasion percent. You'll notice that at 50% invasion, the median value, which is represented by the dark black line, is higher than 0 or 100%. Now, if we also look at what's called the interquartile range, which is represented by the box itself, we notice 100% has the smallest range, which means it has the lowest variation in abundance per plot. Visually, so what do our results mean, and are they significant? Visually, while there might be a slight trend of decreasing insect abundance across insect levels, this difference is not significant. We used an ANOVA to assess how insect abundance vary across different levels of invasion. Farther, while we may see greater variation in insect abundance at lower levels of invasion, the main takeaway is that invasion abundance does not significantly impact insect abundances. This is a highly interesting finding, as the number of insects and generally the same orders are found at high levels of invasion where native plants are lower in abundance, as in low levels of invasion where native plants are higher in abundance. Because insects compete directly with cattle for native forage and avoid invasives during this time of sample collection, this could add farther stress to native plant communities, potentially affecting our long-term stability and usage of our rangeland communities. As stated earlier in this presentation, invasive species have the ability to upend and damage native ecosystems. So it's important to understand their consequences across ecosystems, including insects. Broadly, this research furthers the available data on the impact of invasion with regards to our critical rangeland communities. I'd like to thank my graduate mentor, Morgan Frost, as well as my PI, Dr. Sally Kerner, for their support throughout this project. I'd also like to thank my fellow undergraduate researchers for their help throughout this project. And additionally, I'd like to thank my sources of funding, which are ERSCO, the UNCG Biology Department, and the USDA. This concludes my talk. Thank you all for listening.
All righty, thank you so much. All right, big round of applause for that one too. And I realize I skipped applause for Natalie as well. So an applause for all three of our presenters. Thank you so much. So now we are going to move on to Q&A. So I believe we have until 10:50 for questions and answers. You are welcome to raise your hand and I can let you know when to unmute, or if you would prefer to put your questions into the chat, we can do that as well. And I will read your question out loud for the sake of the recording and have folks answer. And I have a question as well, but I'll give folks a chance to put theirs in first. All right, I'm not seeing any in chat, so I'll go ahead. I have a question for you, Zachary. I am curious how you determined invasion percent, especially for the middle half when you were looking at your two charts in the middle of your presentation.
Yes, so I'm sorry, is that better? You have headphones too. So we determine invasion percent by plant coverage. So we'd look at each plot and see how much of that plot is covered by the invasive plant. So if it's none or almost none, it's 0%. If it's about half, it's 50%. If it's all or almost all, it's 100%.
Thank you.
I'm sorry, what did you say?
Oh, I said thank you.
Okay, great. Does that make sense?
Yes, it does. Thank you.
Okay.
All right, I have a question from Rosalie, who says, great talks, everyone. Natalie and Zachary, do you think insect species richness would change more than insect family by treatment or still be pretty unaffected? I guess, Natalie, do you have a response for that one? I can read it again too, if you'd like.
Sorry, I'm just trying to understand exactly what the question is asking. Do you think insect species richness would change more than insect family by treatment? Okay. So I personally think, based on the research, that species richness would change more when it came to treatment. I think it would be very similar. It's hard to say because the orders and family results were actually insignificant. But when you saw the difference between each community or each treatment plot, that result was significant. So I think I would say that when you're looking at the communities as a whole, then that one would be more effective since that was the result that we got more data behind or more support behind, if that answers the question.
See. Alrighty. And then I have another question for y'all from Jenny. How do your findings and results help? That might be a typo for your approach, but I'm going to read it as it is. How do your findings and results help our approach to different aspects of development and infrastructure in the environment and sustainability, including agriculture, urban development, soil ecology, etc.?
So I can answer part of that. In terms of agriculture, cattle prefer native forage, and so if the insects, like especially Orthoptera, are eating the native forage as well, and they're not eating the invasive forage, you're going to have a decrease in cattle productivity. As far as urban development, I don't think there's quite that much urban development out on rangeland. It's pretty remote. You know, you might have a few farmhouses, but there's not a lot. There's not a lot of like urban construction going on. And soil ecology, that's probably out of the scope of my research.
Yeah, just to add on to that, I do think that it all really goes hand in hand as far as urban development. I mean, they're always trying to take over a lot of natural areas. So especially when it comes to the humans trying to make longleaf pines more, I guess, applicable to our lifestyle, the fire suppression really comes into play with that a lot. But the longleaf pines really need this fire in order to be healthy in their own way. So I think just our ideas on developing and changing these environments for our own use has to do a lot with our research, because we're really trying to help restore these natural areas. So you have to kind of take both into consideration in doing so.
All right. And I have a question for Jenny, but again, I'll see if anyone else has questions first. And I just want to let Zachary and Natalie know I also share an office, so I know that struggle with the echoing microphone. All right. So my question for Jenny, how did you pick the books that you looked at, or the authors that you looked at specifically? And you may have covered this a little bit.
Yeah, so the authors that I had for Bill Gates and John Muir were from my class, Reading Environment. And those were the two pieces. We had to pick two pieces from that class. And those were the two that I chose. And I wanted to choose another two kind of outside, having like a different perspective in both environment and sustainability. And I chose two that I didn't really know a lot about until I actually went and searched. I did Google search, and I found that a lot of the books were still books that, you know, were mainstream and that we all know. So I really had to dig a little deeper into finding Mathai and Kimmer. And those were kind of the two books that I wanted to investigate and analyze.
I had a little question just to like get your opinion, Jenny. How do you feel about, I've obviously reading in the environment is very important. I took that class really recently and I've been trying to read more environmental books. How do you think we could maybe try to make younger kids get really into combining reading in the environment, maybe with more children's books, or making them more accessible to people from all backgrounds?
Yeah, children's books have a very big impact in how we adults and even children see the environment, from Dr. Seuss and the Giving Tree, kind of those different perspectives in kind of preserving the environment, or should we take from the environment. But I think that there are various, even more books from people of color, kind of targeting kind of the audience of children, and kind of having these introducing these concepts about the environment, but also how it connects to their lived experience. And I know that there are various different books written by many beautiful different authors, and kind of increasing that accessibility and to those books is, through libraries, through in our curriculum, even when we're talking about science, having those around and kind of being able to provide that to children, libraries, and schools again.
As a librarian, I am all for that response. All right, so I don't see any more questions in the chat. Again, you're welcome to put them directly into the chat, or if you would like to unmute and ask a question, you are also welcome to do that for our attendees. I don't see any hands raised. I'm always that person who is furiously typing my question when everyone stops asking questions, so. All right, and I have a question, I guess, for Natalie and Zachary. So I know, Natalie, you mentioned you used R to do analysis, and I'm assuming you might have used that for your charts and your graphs as well. The question I want to ask is, what was your experience with that? But that's a very broad, vague question. Had you used R before this, or was this something that you used specifically for this project?
I had not used R before this, so I was really kind of learning on the job. It's something that I know a lot of ecologists use, so it was great for me to get that experience, and I'm using it in another one of my biology classes this semester, which is really great, but it was very new to me, and Dr. Kerner and my grad student, Alyssa, that I'm working with, helped me a lot with that. So I know it a lot better. I still have a lot of work to do on it, but yeah, it was not too horrible.
So yeah, for me, it's kind of the same thing. I'd never used it before, working in the Kerner lab, but all of our wonderful grad students here actually put together a course for our lab to learn R. So once I went through that, I kind of got the basics down, and then they've worked with this type of data before, so you can always go to them for additional help.
That's awesome. It's pretty similar to my own experience with R as well in undergrad. Jenny, I guess I have another question for you as well. I don't see any more in the chat, but again, if you're typing, please keep typing. Oh, cool. Rosalie asks, Natalie and Zachary, do you guys have any favorite insects? And then I'll come back to my question for Jenny afterwards.
Yes, I do. I love looking at the insects. So the spiders kind of freak me out, but the specific spider, the jumping spider that I actually showed a picture of in my presentation, is my favorite spider because it's really cute. Actually, it has like really big eyes, but like not in a creepy way, and the colors are actually really pretty. It can be like kind of iridescent a little bit, like it changes color in the light, and I think that's really cool. And I just think the jumping spiders are neat. And we also get a ton of leaf hoppers, which I didn't show, but they're just like little bugs that you can find on the leaves, and they like munch on the leaves, and they're also very cute. So those are my two favorites.
Yeah, I think I would say I do. Leaf hoppers are definitely one of my top favorites as well because they're super cute. But we also had one grasshopper that he's the biggest one we found, way bigger than any other ones. I didn't include a picture of him, but we named him Little Tim, and so he is now one of my favorite insects.
I love that so much, a named thing. All right, and it is 10:50, but I think we have like 30 more seconds. So Jenny, I would be interested to see if you had any plans to look at any kind of quantitative like numerical data or some other, you did a lot of like qualitative and looking at author experiences. And I know a lot of times folks like to combine those quantitative and qualitative. So I was wondering if you were interested in doing that in future work at all.
Yeah, actually in part of my research, I did look at the impact, the kind of statistics and how many authors there are in BIPOC and Black and white and men and women. That was included in my research. But I'm also interested kind of the quantitative side of how like the difference in values and beliefs and like certain kinds of endorsement and pro-environmental beliefs between different groups. I think that would be a very interesting thing that I would be interested in looking into.
That would be very interesting, like a public opinion and environment.
Yeah.
All right. Cool. Well, thank you all so much. It is 10:51, and thank you to all our attendees for attending, and I guess another round of Zoom applause for our students who presented today. All right. Thank you so much. Have a great Friday.
Thank you all.
Thank you. Thanks, everyone.
Thank you.